Welcome to our session on ICH Quality Guidelines. In this video, we'll walk through the entire framework step by step and understand how these guidelines shape pharmaceutical quality across the globe. Here's our roadmap. We'll begin with the mission of ICH and the Q family map. Then, we'll cover stability and stress testing, analytical validation and impurity control, Pharmacopoeias and biotech guidance, specifications and GMP, and finally the integrated systems approach of Q8 through Q10. Section 1 introduces the mission of ICH and the complete Q family. This will set the foundation for everything else in today's session. The International Council for Harmonization exists to align global requirements so medicines are consistently safe, effective, and high quality. The goals are clear. Harmonize technical requirements, avoid duplication of trials, minimize animal testing, and most importantly, promote public health by accelerating patient access to medicines. Here you see the Q Series roadmap. It's a comprehensive toolkit for quality, starting with stability in Q1, through analytical validation, impurities, pharmacopoeias, biotech products, specifications, GMP, development, risk management, and ending with a pharmaceutical quality system in Q10. Section 2 is all about stability and stress testing, the Q1 framework. The stability package provides a 6-part framework for how stability data is generated and evaluated. It includes general studies, photostability, dosage form extensions, reduced study designs, statistical evaluations, and defines storage conditions for different climatic zones. Q1A lays down the core stability protocol. At least 3 batches must undergo testing. Long-term data is collected over 12 months, accelerated data over 6 months. A stability commitment is required to confirm expiry dating after approval. Q1B introduces a decision tree for photostability. Start with the API under forced degradation. If it's not photosensitive, no further testing is needed. If it is, extend testing to the finished product and packaging. Light impact during manufacturing must also be assessed. Q1C and Q1D provide streamlined testing strategies. Q1C reduces long-term data for new dosage forms. Q1D introduces bracketing, testing extremes like the largest and smallest packs, and matrixing, where factor combinations are tested at different times. Section 3 now takes us to analytics and impurity control, the Q2 and Q3 guidelines. Q2 defines how analytical methods must be validated. Key characteristics include accuracy, precision, specificity, linearity, detection and quantitation limits, range, and robustness. The guideline ensures tests like identification, impurity limits, and assays are reliable. Q3 is divided into 3 parts: organic impurities in Q3A, inorganic impurities in Q3B, and residual solvents in Q3C. All define thresholds for reporting, identification, and qualification to safeguard patients while avoiding excessive testing. Residual solvents are classified by risk. Class 1 solvents, like benzene, are to be avoided. Class 2 solvents, like methanol, can only be used within strict limits. Class 3 solvents, like ethanol and acetone, are low toxicity and less concerning. Section 4 looks at pharmacopoeial harmonization and biotechnological products. Q4 aims to harmonize pharmacopoeial standards across regions. Q4A sets the process, while Q4B defines regulatory acceptance of harmonized texts. Examples include dissolution, sterility, friability, electrophoresis, and endotoxin testing. Q5A introduces viral safety for biotech products. This involves testing and selecting cell lines, validating the virus clearance capacity of the process, and monitoring intermediates to ensure products remain safe. Q5 extends across the biotech lifecycle. Q5B focuses on expression constructs, Q5C on stability, Q5D on cell substrate qualification, and Q5E on comparability after manufacturing changes to ensure safety, quality, and efficacy are maintained. Section 5 is about specifications and GMP under Q6 and Q7. Q6A defines 2 levels of testing. Universal tests like description, identification, assay, and impurity control apply to every product. Specific tests are added depending on dosage form, for example, dissolution for oral products or sterility for injectables. Q7 extends GMP to APIs. It covers facility design, documentation, validation, change control, lab testing, packaging, storage, recalls, and complaints. Special guidance is also provided for fermentation and cell culture APIs. Section 6 wraps up with Q8 to Q10, the integrated quality paradigm. Q8 is Pharmaceutical Development, embedding quality into design. Q9 is Risk Management, providing tools to assess and control risks across the life cycle. Q10 is the Pharmaceutical Quality System, a management framework that ensures continuous improvement. And that brings us to slide 24. Thank you for watching. I hope this walkthrough clarified the ICH quality guidelines. Don't forget to like, share, and subscribe for more regulatory and pharmaceutical insights.